Hi, everyone. It's great to be with you. I'm sorry I can't be there in person. Thanks for your patience as I beam in from the central part of the United States today. What I thought I would do is to add to what you've already heard from Michael Levin and from Jakob Hanna about the amazing properties of cells derived from embryos, and talk a little bit about the amazing processes that occur during normal embryonic development, and then talk briefly about the amazing resilience of the early embryo and some of the implications. I'll admit that I'm not an expert on Buddhism. My own tradition is the Christian tradition, and yet I think there are a lot of similarities with some of the questions that are raised by embryonic development with regard to the nature of persons that arise both in Buddhism and in Western religious traditions. But let's start with the science. Where are we headed? First, I want to make the case that understanding embryonic development is an opportunity for wonder. And then I want to go on and point out that understanding embryonic resilience is a starting point for tackling the intersection of technology and human identity. And this raises questions for Buddhism as well as other religious traditions. Well, this is me a long time ago. This is my son, John. And if you're a parent, you'll appreciate the amazing processes of embryonic development. When you hold a baby in your arms, you can't but be struck with the remarkable reproducibility of development. How all the digits seem to be formed in this amazing way, all of the other parts of a baby. It's remarkable. And it's an opportunity for wonder. As a developmental biologist, understanding how an embryo is built provides even more opportunities for wonder. We all started as a one-celled zygote, as you see on the upper left. Then, as shown by the white arrow, creatively enough, complicated things happen, and then you get something really complex. This is my son, my younger son, Christopher, a long time ago. And these processes that result in the construction of an embryo are absolutely staggering. In fact, a human infant has about 5 trillion cells, and that's a large number, but we all began as a one-celled zygote. That's the set of processes that fascinate developmental biologists like myself. One of the things that we understand about embryonic development in the Western tradition goes all the way back to Aristotle. It turns out that people in the Eastern tradition were also onto this idea. It's the notion of something called epigenesis. This is the notion that complexity arises through the interaction of simpler parts. And you've already heard about this from Michael Levin and from Jakob Hanna. Well, what's going on as that emergent process is carried out through embryonic development? Well, one thing that's happening is that parts are becoming different, and developmental biologists call that differentiation. This involves changes in gene expression, but also, very dramatically, the ability of cells to communicate with one another. But that's not enough. You also need something called morphogenesis, and this is what my own lab studies. This is the notion of building the body from the Greek word morphe, which means form. Now, it's certainly true that genes are arrayed in a number of networks called gene regulatory networks, in fact. And you can think of these as control systems that make it possible for certain kinds of modules to be deployed during embryonic development. So in that sense, genes provide the information to make a set of protein switches. And these are often regulated by communication. And these switches can be really complicated. They can activate the expression of genes that lead to the production of new proteins. But we can also think of the opposite thing occurring. In certain cases, certain genes stop being expressed. And a lot of effort's gone into understanding these gene regulatory networks. But those are not enough. And to show you why that's true, I don't know how many of you have children. I have 2 boys, as I've already pointed out. Well, they played with Lego bricks. And Lego bricks come in different colors, sizes and shapes as you see on the left. But a pile of these different kinds of bricks is not enough. You have to assemble them into some sort of structure so that you get not the pile on the left, but the intricate structure that you see on the right. And that's what morphogenesis does. And you've already heard a bit about that from Michael Levin. Well, that's what my lab studies. We use a little nematode worm called Caenorhabditis elegans to do this. And here's just one example of the set of movements that my lab studies using sophisticated microscopes. So this is a living embryo, and you're watching cells move, and you don't need to know what's going on except to say that the skin is covering over the interior of the embryo. And we can genetically engineer these embryos so that they make glowing proteins that we can follow with specialized microscopes, like the protein on the right, which is something called alpha-catenin. These complicated cellular dance moves are part and parcel of morphogenesis. Well, let's then take a whirlwind tour of human development, because although this is a little bit out of order, I think in terms of what you've already heard at this conference, it's probably worth everyone understanding the technical words that are used to describe the embryo at various stages. First is fertilization. Fertilization is an incredibly contingent process where one sperm unites with an oocyte, an unfertilized egg. In fact, estimates are that a single sperm has a 1 in 250 million chance of fertilizing an egg. But once this happens, it sets in process a series of very complicated physiological changes in the fertilized egg that ultimately lead it to prepare for its first division, to go from 1 cell to 2. And that process begins with this. This is a zygote, and you can see that there are 2 nuclei here. They contain genetic information, one from the sperm and one from the oocyte, and they've come together in preparation for the first cell division. So that's called a zygote. The zygote then divides in a process called cleavage. And in humans, in typical humans, produced the old-fashioned way, this happens as the embryo is passing down the fallopian tube or the oviduct, and you see a human embryo doing this here. This is a human embryo with 4 cells. This happens during the first several days of human development. So these early cell divisions are called cleavage, and here is cleavage in a mouse embryo. Mice look strikingly similar to human embryos at this stage of their development. And as I play this movie, you can see that these cells are dividing from 2 to 4, from 4 to 8, and then a cellular glue is produced on the surface of these cells, and this leads to an event called compaction. Eventually, the interior of the embryo fills with fluid, as you see towards the end of this movie, as the movie is looping. Now, there are a lot of complicated things happening here. Inner cells become different from outer cells in terms of their differentiation, a word we've already used. Eventually, you get this. This is a blastocyst, and a blastocyst has these very specialized cells on one end called the inner cell mass. That's what eventually is going to make the baby. And you've already learned from Jakob Hanna that these are incredibly developmentally flexible cells. They are pluripotent, and when they're cultured, they can make pluripotent stem cells. Well, at this stage of development, the embryo attaches to the uterine lining of the mother and continues development. One of the things that happens is that the embryonic body plan begins to emerge during a very complicated process that I've studied throughout my career called gastrulation. This is a human gastrula with some of the membranes removed. And you can see that there's a dark streak here down the middle. That's called the primitive streak. And that forms by really complicated movements of cells moving towards what becomes the middle part of the embryo as the basic body plan emerges. Well, here's gastrulation in some genetically engineered mouse embryos. They were expressing fluorescent proteins, some magenta and some green, and we can follow the embryo as cells perform these movements. And you can see that the embryo gets much longer from top to bottom. That's the anterior to posterior or head to tail axis of the embryo forming. In a mouse embryo, this involves thousands of cells. And you can see that this is a really complicated bit of cellular choreography. Well, eventually, a flat sheet rolls up into a tube during what's called the neurula stage as the central nervous system forms. And at this stage, we all basically looked like a burrito. Sorry for that Western food analogy, but you get the idea. And here's neurulation happening in axolotl embryos. So these are salamanders or related to salamanders. And you can see that there's a flat sheet of tissue that rolls up and eventually covers over the surface. I'm going to loop through this again. Here's this flat sheet called the neural plate, and you can see that there's a complicated bit of morphogenesis happening as the central nervous system forms. Well, following that stage, organ systems begin to arise, including the brain and other tissues derived from that tube that formed during neurulation, but lots of other organ systems as well coming from other parts of the embryo. All of this is happening within the first 8 weeks of human development. And it's this sequence of processes that fascinates developmental biologists. This is all happening before the mother first feels the fetus, which typically happens at about 16 to 22 weeks of development. Remarkable. What can we say about this? Well, first we can say that we all started as a one-celled zygote, as you see on the upper left, and we all ended up as a baby, as you see my son Christopher on the lower right. It's the same organism that is present at all of those stages, at least that's what we can say biologically. And what's fascinating about this is that this is a massive construction project in which the embryo builds its own body. And the scientific questions that arise from observing this process are many, and that's what fascinates developmental biologists. Well, what can we say about embryonic development in dialogue with Buddhism? I really want to encourage you to review your notes from Robert Kritzer's talk. He's done some really important work in this area, especially interacting with ancient Buddhist and Tibetan medical texts. But one of the things we certainly can say is that there's no Buddhist view of embryonic development as if there's some sort of monolithic approach here. One other thing that we can say makes it somewhat challenging here is that Buddhist ideas in general predate modern embryology. Here's an example, the Gandhabba. How do we unite the material elements that we see in an embryo at or near conception with immaterial elements? How should we think about that? Or here's a quote from the Great Causes Discourse Mahanidana Sutta. If consciousness were not to descend into the mother's womb, would name and form a special kind of idea in Buddhist texts take shape in the womb? In the Garbhavakranti Sutra, there are remarkably prescient descriptions of development, and I think Dr. Kritzer has really done a good job of fleshing those out. But here too, there's discussion of winds. Somehow these are layered onto descriptive embryology. And what do these mean metaphysically? In general, I think there's an underlying context that involves suffering. Embryos are evidence of dukkha and agents of samsara. And again, I want to point you to some work that Dr. Kritzer has done, really great work. And I want to recommend this one reference here. My colleague John Wallingford, a developmental biologist, has also done some interaction with Dr. Kritzer's work that I can point you to. All right, so that's a little bit of embryonic development and some of the issues that are touch points with Buddhism. How about the second thing that I wanted to talk about, the resiliency of the embryo? Well, to illustrate that, I want to draw your attention to this gentleman. This is Hans Striesch, a German embryologist at the end of the 19th century, and he worked with embryos of an organism that I've worked on, the sea urchin. And Triche found a way to separate the cells of an embryo when it only has 4 cells and to culture the individual cells separately. What Triche found was, remarkably, each of those individual cells can give rise to a perfectly proportioned baby sea urchin, a sea urchin larva. And this is indicative of characteristics that many embryos have, a remarkable ability to self-organize following injury or experimental manipulation. They're resilient. Now, this so flummoxed Hans Triche that he abandoned embryology and he became a philosopher. But we know that this is a common event during embryonic development in many animal embryos, including humans. And so these beautiful monozygotic or so-called identical twin girls are evidence of that. That twinning due to embryo splitting can occur, and we can infer when it occurs in humans. It can actually occur pretty late, until right before gastrulation. Now, I want to talk about a consequence of this resiliency of the embryo, and that involves something called chimeric embryos. To begin our discussion of chimeric embryos, I want to explain a mind-blowing experiment to you. In mouse embryos, where this was originally done, it's possible to take multiple embryos prior to the time that the cells make this cellular glue that causes the cells to adhere to one another very tightly. So you can push 3 different embryos together, and here you see 3 different mouse embryos on the left. When they start making this cellular glue protein, they will unite into a single embryo, as you see in the middle. And then on the right, you'll see that there's a single blastocyst, and the inner cell mass is very big here, but eventually the proportions of the embryo sort themselves out. And this leads to what's called a chimeric embryo, an embryo derived from embryos that have different genetic constitutions. You can also do this later by injecting those specialized cells, the inner cell mass cells, into a blastocyst. And here's what you get when you do this kind of an experiment in mice. If you start with embryos derived from mice that have 3 different genetic constitutions indicated by their fur color, you can see that you can get an embryo that has islands of tissue derived from the starting embryos. And so you get a chimeric embryo. And it turns out that this also works in monkeys. So in this particular example, which made the cover of a well-known science journal, in 2012, 6 different embryos were combined to form a single monkey embryo. And you can see you get a very cute baby monkey. Now, there's good evidence that this happens spontaneously at times, so there are humans that seem to be genetic mosaics. All of this is to say that the resiliency of the embryo makes this possible. Well, how should we think about this in terms of technologies? Well, it turns out people are interested in making human-animal chimeras. The idea here is to use genetically engineered pig embryos that have certain immunological properties and to introduce human cells into these embryos. And if these embryos are produced in a particular way, then a particular organ will be predominantly populated by the human cells, and that's the red cells that you see in this particular experiment from the Ispasua Belmonte lab. So the human cells here are colored red. Why would we want to do this? Well, this might be a way to culture tissue that could be used in organ transplantation. Now, this raises all kinds of questions. One of the questions that you can see is that there are some red cells perilously close to the brain of this particular chimeric embryo. Raises lots of questions. But the idea here is that the human cells would form an organ. This has been tried with human monkey chimeras. This raises lots of questions about the status, moral status, and the biological status of these chimeric embryos. Well, how should we assess chimeras? Well, In the US, the National Academy of Sciences said these kinds of studies could produce creatures in which the lines between human and non-human primates are blurred, a development that could threaten to undermine human dignity. This would be especially problematic if it involved brain tissue. Now, I think there are alternatives that are available. Induced pluripotent cells, something that you learned about at this conference, are maybe a good alternative technology. Well, how do we think about this in the context of Buddhism? Well, how manipulations of embryos, including intentional chimeras, should be regarded in terms of the Four Noble Truths of Buddhism, for me, is a major challenge and something I hope to learn about more at this conference. There are a lot of other questions at the interface between Buddhism and technologies as applied to embryos. But that's enough to get us going for the discussion today. Thanks very much.